For centuries, the Turin Shroud has remained one of the most mysterious artifacts in human history. Preserved in the historic city of Turin, the ancient linen cloth has fascinated historians, scientists, archaeologists, and religious scholars alike. The faint image visible on its surface has inspired countless debates, investigations, and theories. Some believe it represents one of the most important relics ever preserved. Others view it as a remarkable historical object whose true origins remain uncertain. Despite decades of research, many questions surrounding the cloth have never been fully answered. How old is it? Where did it come from? How was the mysterious image formed? And perhaps most importantly, what hidden information still remains locked within its fibers? For many years, researchers focused primarily on the visible features of the cloth. They examined the image, they analyzed stains, they studied the weave of the linen, they conducted chemical tests, they used photography, spectroscopy, microscopy, and numerous other scientific techniques. Each investigation produced valuable information. Yet one area remained largely unexplored until modern technology made it possible: DNA. Hidden deep within the fibers of the Turin Shroud were microscopic biological traces, accumulated over centuries. Tiny fragments invisible to the naked eye, particles left behind by people, plants, microorganisms, and environmental exposure. For generations, these traces remained beyond the reach of science. Then, genetic technology changed everything. As DNA analysis became increasingly sophisticated, researchers realized that ancient artifacts could contain remarkable biological records. Objects handled by human beings often preserve microscopic evidence of those interactions. Skin cells, hair fragments, plant material, microorganisms, all can leave genetic signatures behind. Even centuries later, traces may remain detectable. This realization inspired scientists to take a closer look at the Turin Shroud. The project was ambitious. Ancient DNA research presents enormous challenges. Biological material degrades over time. Environmental contamination complicates analysis. Modern handling can introduce additional genetic material. Researchers therefore approached the investigation with extreme caution. Every sample had to be carefully collected. Every result required careful verification. The goal was simple: determine whether meaningful biological information still existed within the cloth. What they discovered exceeded expectations. Microscopic analysis revealed that the Turin Shroud contained a surprising variety of biological traces. These traces came from multiple sources. Some were associated with plants, others appeared linked to microorganisms. Most intriguing of all were genetic signatures connected to human populations from different geographic regions. The findings immediately attracted scientific attention. For centuries, the Turin Shroud had traveled through history. It had been displayed publicly, transported between locations, examined by researchers, viewed by pilgrims, handled by countless individuals. The genetic evidence suggested that many of those interactions had left microscopic traces behind. Researchers identified DNA associated with populations from Europe. This was not surprising. The cloth has spent centuries on the European continent, but additional findings proved more intriguing. Some genetic signatures appeared connected to regions beyond Europe, areas of the Middle East, parts of the Mediterranean, and other locations historically linked through trade, travel, and cultural exchange. Scientists immediately recognized the significance of these discoveries, not because they proved the origin of the cloth, and not because they identified any specific individual. Rather, the evidence suggested that the Turin Shroud possessed a far more complex biological history than many researchers had expected. The cloth appeared to have interacted with people from numerous regions over long periods of time. This observation aligned with historical possibilities. Throughout history, important religious and cultural artifacts often traveled great distances. They passed through cities, crossed borders, moved along trade routes, and attracted visitors from many different backgrounds. The Mediterranean world in particular served as a crossroads connecting Europe, Asia, and the Middle East. Objects moving through such networks naturally accumulated traces from diverse populations, yet some aspects of the findings remained surprising. Researchers also detected genetic material associated with numerous plant species. The presence of plant DNA opened another avenue of investigation. Plants grow in specific environments. Their genetic signatures can sometimes provide clues regarding geographic history. Scientists therefore began comparing the detected plant material with known regional distributions. Some results suggested links to areas outside Europe. Again, caution was necessary. Plant material can travel through wind, trade, transportation, and human contact. The findings did not prove the cloth had originated in any specific location. However, they reinforced the broader conclusion that the artifact possessed a long and geographically complex history. Meanwhile, another group of researchers focused on microbial DNA. Microorganisms exist virtually everywhere. Different environments support different microbial communities. As objects move through various locations, they may accumulate distinctive microbial signatures. The Turin Shroud proved no exception. Analysis revealed a remarkable diversity of microbial traces. Some were consistent with European environments. Others appeared linked to regions farther east. Scientists emphasized that interpreting microbial evidence remains challenging. Microorganisms spread easily. Environmental contamination is common. Definitive conclusions are difficult. Nevertheless, the findings contributed another piece to the puzzle. The biological record preserved within the cloth appeared extraordinarily diverse. As news of the research spread, public interest grew rapidly. Media reports described hidden DNA traces, ancient genetic evidence, unexpected discoveries. Some headlines were dramatic, others were cautious. Scientists themselves urged restraint. The findings were important, but they did not solve the mystery of the Turin Shroud. This distinction mattered greatly. Scientific evidence must be interpreted carefully. DNA can reveal patterns. It can identify biological relationships. It can provide clues regarding environmental history, but it cannot automatically answer every historical question. Researchers repeatedly emphasized what the evidence actually demonstrated. The cloth contained genetic material from multiple sources. Those sources reflected a long history of interaction with different people and environments. Beyond that, conclusions became increasingly uncertain. Still, the discoveries represented a significant scientific achievement. For the first time, researchers could examine aspects of the Turin Shroud invisible to previous generations. Microscopic biological traces had become valuable historical evidence. The cloth was no longer viewed solely as a visual artifact. It was also a biological archive. Every century of exposure, every journey, every interaction may have left traces preserved within its fibers. This realization transformed how many scientists approached the artifact. Instead of focusing exclusively on the image, researchers began viewing the entire cloth as a source of historical information. The fibers themselves contained data. The biological residues contained data. Even microscopic particles trapped within the weave could contribute valuable insights. The possibilities seemed enormous. As investigations continued, researchers developed increasingly advanced analytical methods. Improved DNA sequencing technologies allowed examination of ever smaller samples. Enhanced contamination controls improved reliability. New computational tools helped identify genetic patterns hidden within complex datasets. Each advance expanded scientific understanding. Yet the more researchers learned, the more fascinating the mystery became. Because the genetic evidence hinted at something larger, something beyond a single artifact. The Turin Shroud appeared to preserve a record of human interactions spanning centuries. People from different cultures, different regions, different periods of history, all may have contributed to the biological traces now being studied. This perspective fascinated historians. The cloth had effectively become a microscopic record of human movement and contact, a biological witness to history itself. But perhaps the most surprising discoveries were still ahead. As scientists continued analyzing the hidden DNA traces, new findings began emerging, findings that would generate intense discussion within both scientific and historical communities. Because buried within the growing collection of genetic data were clues that researchers had not anticipated, clues that raised entirely new questions about the Turin Shroud's extraordinary journey through time. And those questions would soon become the focus of one of the most intriguing investigations ever conducted on the mysterious cloth. Researchers uncovered the first layer of genetic evidence hidden within the Turin Shroud, traces of human, plant, and microbial DNA that suggested a long and complex history of contact across different regions and centuries. But as the investigation continued, scientists began to realize something even more unusual. The deeper they analyzed the data, the more patterns began to appear, and those patterns were not easy to explain. The Turin Shroud was revealing a biological record that no one had expected. Not just random contamination, not just modern interference, but a layered and genetic footprint that seemed to reflect multiple historical phases of contact, some ancient, some medieval, and some surprisingly recent. This raised a critical question. How could a single artifact carry such a wide range of genetic signatures? To answer this, researchers divided their study into categories, separating human DNA, environmental DNA, and microbial DNA into distinct groups for analysis. What they discovered next intensified the mystery. The most striking findings came from human genetic traces. Scientists identified multiple genetic profiles embedded in microscopic residues on the cloth. These profiles were not complete genomes, far from it. Instead, they were fragmented DNA signatures, too degraded for full identification, but still informative enough to suggest regional origins. Some markers appeared consistent with Southern European populations. Others showed similarities with Near Eastern genetic patterns. A smaller subset hinted at broader Mediterranean diversity. At first, this seemed logical. The Turin Shroud had traveled extensively through Europe over centuries, so mixed genetic signals were expected. But what puzzled researchers was the distribution pattern. The DNA traces were not evenly spread. Certain sections of the cloth showed higher concentrations of specific regional markers. Other areas showed almost none. This uneven distribution suggested repeated handling in specific contexts rather than random exposure. One research group proposed a hypothesis. The cloth may have been exposed to different populations during distinct historical phases. For example, early contact in one geographic region, later movement through Mediterranean trade routes, much later exposure in Europe during public display periods. This layered interpretation began gaining attention because it explained something important. The genetic data did not look like a single moment in history. It looked like multiple chapters. As sequencing technology improved, researchers revisited older samples. This time, they used more sensitive detection methods capable of identifying extremely rare DNA fragments. That decision led to a surprising breakthrough. Among the vast mixture of genetic material, scientists detected rare markers that did not match common modern reference populations. Instead, these markers appeared closer to ancient or isolated genetic lineages. This did not mean they belonged to any specific known ancient person, but it suggested something important. The cloth may have interacted with populations that no longer exist in the same genetic form today. This possibility immediately sparked debate. Some scientists argued that degradation and contamination could explain the anomalies. Others believed the patterns were too consistent to ignore. A few researchers cautiously suggested that the shroud might preserve genetic echoes from very old human interactions, possibly predating its documented European history. However, no one could confirm this definitively. The data was suggestive, not conclusive. Still, the idea changed the direction of the research. While human DNA attracted the most attention, microbial DNA turned out to be equally revealing. Microorganisms behave like environmental timestamps. Different environments leave different microbial signatures. So by studying them, scientists can reconstruct parts of an object's exposure history. On the Turin Shroud, microbial analysis revealed something unexpected: a layered microbial ecosystem. Some microbial strains matched long-term storage conditions typical of European churches and museums. Others suggested exposure to warmer, more humid climates. And a few strains appeared consistent with environments found in the Middle East. This combination was puzzling because it implied the cloth had not remained in a single stable environment throughout its history. Instead, it may have moved through multiple climates and storage conditions over centuries. One microbiologist described it this way: the shroud is not just carrying DNA, it is carrying environmental memory. This idea became central to later discussions. As the results became more complex, scientists faced a major challenge. How much of the DNA was historical, and how much was modern contamination? This question is critical in ancient DNA studies, because even a single modern skin cell can alter interpretations. Researchers therefore re-examined every step of the sampling process. They tested equipment contamination, they checked handling records, they compared multiple independent laboratories. Despite these precautions, uncertainty remained. Some DNA clearly came from modern handling, especially from known exposure during research and public display, but other fragments were far too degraded to be recent. These ancient fragments could not be easily dismissed. This created a scientific tension. Two possibilities existed. The DNA represents a genuine historical record of the cloth's long journey. The DNA is an incomplete mixture of contamination accumulated over decades. Both explanations were plausible. Neither could be fully proven. As data accumulated, researchers attempted to map the genetic findings geographically. They created models showing where different DNA signatures most likely originated. The results were surprising. Instead of a single origin point, the model suggested multiple overlapping regions: Western Europe, the Mediterranean basin, parts of the Near East. This pattern aligned with known historical trade and religious roots, but it also introduced a deeper question. Why would an artifact show such broad geographic interaction unless it had traveled extensively or been repeatedly exposed to diverse populations? One historian noted, "Very few objects in history have passed through so many hands across so many cultures. The shroud appeared to be one of them." As discussions continued, a new working theory emerged within parts of the scientific community. It was not a claim of authenticity, nor was it a claim of forgery. Instead, it was a neutral hypothesis based purely on genetic data. The Turin Shroud may be a multi-phase artifact, meaning its biological and environmental history is not tied to a single moment, but to a long sequence of events. According to this idea, the cloth may have originated in one region, traveled through multiple cultures, been preserved, handled, and reinterpreted over centuries, accumulating genetic traces at each stage. This would explain mixed DNA signals, geographic diversity, microbial layering, uneven distribution patterns. However, this theory did not solve the biggest mystery. It still did not explain the origin of the image itself, and it still did not answer whether the cloth's earliest history matched its later traditions. As research progressed, scientists openly acknowledged a difficult truth. DNA alone cannot reconstruct complete history. It can provide clues, it can suggest patterns, but it cannot tell the full story without context. One geneticist summarized it clearly. "We are reading fragments of a very long story, but we are missing most of the pages." This statement became widely quoted in academic discussions, because it captured the reality of the research perfectly. The Turin Shroud was revealing information, but not complete answers. At the end of the second major research phase, scientists reached a turning point. They had identified multiple human DNA signatures, detected diverse microbial ecosystems, found plant-based geographic indicators, observed layered contamination patterns, suggested multi-phase historical exposure, but they still lacked one crucial element: a clear timeline. Without a timeline, interpretation remained uncertain. So researchers decided to shift focus again. This time, they would not only analyze DNA, they would integrate genetics with radiocarbon data, textile analysis, and historical documentation. The goal was ambitious: to build the most complete scientific model of the Turin Shroud ever attempted. And what they were about to uncover next would challenge several long-held assumptions about the artifact's past.